for the opportunity to come and talk this afternoon. I'm an endocrinologist, so although I do see patients with tumours, most of my patients don't die from tumours, although I was very inspired by Andrew's talk today. I'm going to talk this afternoon about monogenic diseases, and these, we might think of these as being rare, but if you actually look at the numbers, 1 to 2% of Australians have a clinically significant monogenic disease. When I was thinking about this afternoon, and I hope that this doesn't bring home any grief to anybody in the audience, I thought that in the audience here we're going to have 4 people who are carriers for hemochromatosis, and one person who is, yes, thank you, my husband, and one person who's going to have cystic fibrosis carriage case. So the questions that I was asked to think about this afternoon was whether massive parallel sequencing would be clinically useful in monogenic disease. And the 2 things that I'm going to talk about this afternoon is to talk about identification of hitherto unidentified genes, and also to be able to screen new known genes more efficiently and cost effectively. So to talk first about what a monogenic disease is, they are usually rare, as I said, about 1 to 2%, but these are conditions in which our lovely friend Gregor Mendel defined inheritance according to the garden peas, and everyone will remember that from their year 9 genetics. So remembering this, you can see that the disease is passing from generation to generation according to certain patterns, either recessive or dominant, autosomal or X-linked. So within a family who's got Mendelian disease, everyone in the family will usually have the same mutation. But between different families, they may not necessarily be the same mutation, but they are usually in the same gene. Mapping monogenic disease by linkage in most of the 90s was really actually quite successful. But to be successful at mapping it, you really need to have a number of informative meioses, and that depends on how many generations are affected and how many affected individuals there are within families, and how well there is penetrance. If you're actually carrying the genotype, how likely you are to actually manifest the phenotype. And I put there at the bottom some of the things that I see as an endocrinologist, which are monogenic diseases, and I put them up for specific purposes. So Marfan syndrome, everyone with Marfan syndrome has got a mutation with FBM1, but almost in every single case, it's unique to the family. And fibrillin is an extremely large gene. So there's no point that you can just go and look at. You've got an awful lot of gene, I think something like 35 or so exons that you have to look at. Osteogenesis imperfecta, most of it in COL1 alpha 1 and COL1 alpha 2, but there's now 18 known genes for OI. And congenital adrenal hypertrophy, at least 3 or 4 common, but a number of other uncommon problems in steroid metabolism that you have to think about with these conditions. Fibrodysplasia ossificans progressiva was actually mapped in my husband's laboratory in Oxford when I was on maternity leave. And what they found was that there was a recurrent mutation in this particular bone morphogenic protein receptor that caused this condition. This is a terrible, terrible illness where people gradually get encased by bone. So the bone formation is triggered by inflammation, things like having a vaccine or having your teeth pulled out. And these children, or adults really, usually die in their 20s or 30s from pneumonia or malnutrition because of starvation, because they can't eat, because their jaws fuse. So it's a really bad condition. You can tell who's going to have it if you're extremely astute by noticing this almost bunion-like lesion that occurs in the big toe. And that's essentially all you can see at birth. But as time goes on, this terrible encasement by bone occurs. It took decades to have enough families who had enough children, who survived long enough to have children, to be able to map this. And eventually, it came down to this unaffected individual and those 2 affected individuals that said it had to be somewhere in this region. And people then went on and did the sequencing and identified the mutation. The relevance of that was that nearly all cases have this. So you can tell whether somebody's going to have this at birth. You don't have to just look for the big toe and you can watch what goes on. Once you know which gene it is, they've been able to determine the structure of the protein, and this has led to identification for potential treatment and trials for this awful condition, which are going to be happening here in Brisbane in the new year. ARC trial. Last year, we wrote a fairly controversial statement in a review article that was celebrating the 10 years since the publication of the human genome. And what we wrote was that if massive parallel technologies had been available when the search for the FOP gene began, the answer could have been found in 15 weeks. Not in 15 years. So that's a fairly controversial statement, and what I want to do now is to tell you that that statement is not science fiction. This is here and now. So, new gene discovery for massive parallel sequencing. Why do we need to think about new approaches? Why can't we go to the old things like linkage or association? Well, I've mentioned already, if you're looking for linkage, you're looking in families, it depends how big the family is and the number of affected individuals. And if you've got diseases which affect reproductive fitness, such as skeletal dysplasia, very hard to have big enough families. I'm sure you've opened 1 million copies of New England Journal of Medicine, seen lots and lots of GWAS, studies in association. We've got multiple unrelated cases who are with association detect using tag SNPs. Well, these are rare diseases, very hard to get enough numbers. These GWAS take thousands of people to have enough power. And secondly, most of the things we're looking for are really very rare, so they're not going to be picked up at using tag SNPs if the minor allele frequency of the tag SNPs is more than 5%, and many of them are de novo mutations. So if it's a de novo in the family, nobody else is going to be affected, so you can't use linkage. And if you've got a de novo mutation occurring, you don't have a common haplotype, you're not going to pick it up by GWAS because nobody else in the community is going to have the same background to be able to pick it up at. So the question we asked was, can massive parallel sequencing identify genes causing monogenic diseases in the absence of any previous linkage or association data? And the answer is yes. So the first paper we published was submitted in 2010, and what we did here was that we took a family who had what looked like anexectic dysplasia, a form of dwarfism. And they were 2 affected children from non-consanguineous parents. The gene that had been known for anexectic dysplasia was RMRP, and this gene was screened and there were no variants in there. And so we then went on to do whole exome sequencing, and at this point we put on the whole family quartet, although nowadays we'd be much more parsimonious. And what we found, this was again early days, so we didn't have a huge number of variants, about 15,000 per individual, about 500 or so that were novel, hadn't been reported previously, about 200 that resulted in a change in the amino acid, and about 100 of those that were predicted to be damaging. But it's still too many to be able to go forward. So we then added a Mendelian approach to it and asked the question, what was the likely inheritance? So we thought the most likely was a compound heterozygous inheritance, that each child had inherited 1 mutation in the same gene, but in a different place in that gene, 1 from each parent. Although the parents were non-consanguineous, we did look for homozygous just to make sure that that wasn't the case. And the other possibility was that one of the parents had gonadal mosaicism and therefore was passing it on, although they themselves were normal. And when we did this, the only model that fitted was the compound heterozygous one. There were 4 genes where this fitted, but in only one of them was this predicted to be damaging in a gene called POP1. And POP1, lo and behold, was the largest individual protein component of the structure which talks to RMRP, the gene that we knew for anexectic dysplasia. So 4 people, we found the cause, new cause, and what we demonstrated was that anexectic dysplasia is actually a disease where the phenotype can result from mutations in a number of genes along the way, and change the thinking about it. That's in a family. What about sporadic disorders? And here we took 5 individuals who had this condition called multicentric carpotarsal osteolysis. This is a condition that often presents in young children, and it's often mistaken for juvenile rheumatoid arthritis because these children get pain and inflammation. And what happens is, if you look at this carefully, they lose all their wrist bones, all their carpal bones, and they lose all their tarsal bones as well. And it can also progress up the arm from there. About 2/3 of cases also get problems with their kidneys, to the point that by their 20s, they're usually on renal replacement therapy or needing to be transplanted. So we took 5 individuals in this case, and what we did was that we did whole exome sequencing, and we asked a different question. The question we asked was there a novel variant that was carried by all individuals? Or were there a variant that was unique to each individual, but was occurring in a common gene? And when we did this, we found that every individual had a damaging mutation within a 17 amino acids period in the same gene called MAFB. And this is showing here not just the 5 that we did with exome sequencing in bold, but a number of other that we had recruited in the community, showing that in every single case, there's a variant in the same gene. So we now know what the cause is, and my PhD student has now been working out what's actually going on so that we can step in to do something about it. More recently, Adine McInerney-Leo, who's going to be talking later, has, as part of her PhD, managed to find 3 novel genes for short-rib polydactyly syndromes, and these were mapped in each case by only sequencing a single individual. We've become much more parsimonious, and then we've been able to talk to other groups who also have 1 or 2 individuals to confirm our findings. These conditions here are all affecting this sort of cilia structure here. I've put 3 up there, and we found 2 others as well, which I can't tell you about or I'll have to shoot you. Okay, and this has resulted in very important papers in this area here. We've also been able to map autosomal dominant diseases in spondylocostal dysostosis. And when we started off this one, we did the father, 2 affected children, the uncle and the mother. Now, arguably, we could have probably just put on the 2 children and the uncle to have been able to have found this. And I want to talk about this one a little bit in terms of lung adenocarcinoma, because I think this one is really a game shifter for this particular family. In this case, we had a very unusual situation where we've got heritable lung cancer. Grandfather had died of it. Mother had lung cancer. I don't know if she is still alive or not. And then her 2 daughters by separate fathers had also got lung cancer as well. Now, we put on the mother, and again, we could argue about whether we needed to put on the mother, but anyway, that's what we put on. And when we analysed this data in this family, we said it had to fit the Mendelian inheritance, that there was heterozygous carriage in the affected individuals, and the unaffected grandmother had to have nothing, because we knew it had to have come from granddad. Granddad was dead, couldn't get his DNA, but we had healthy grandma. So we knew whatever she had wasn't important in terms of causing the disease. This was a very rare condition, so we said things had to be novel, it had to be a coding mutation of good quality, not due to artefact, and we said it had to be in a conserved region where we look at the genome evolutionary rate profiling, so we know that this base has been maintained down evolution. Something that's maintained is usually a very important base, something that's not maintained may not be quite so critically important. And when we did this, we found the gene. So this is in this particular gene has been associated, not this particular one, but one in the family with somatic lung cancers, and this is all very exciting. So, how many cases are going to... Yes, and I'm going to shoot, so you're going to shoot the others, okay. I didn't think you could read it from there. How many cases are needed to map a causative gene? This depends on the population frequency of the disease, the mode of inheritance, and the presence or absence of consanguinity. So now, if we are looking at a rare autosomal recessive disorder, we can be highly parsimonious, and we can get this even with sequencing just one individual. Because if you have a non-consanguineous family and you pick up 2 rare or novel variants in a single gene, in a single individual, that's highly suggestive of causality. If you've got a second child, you can reduce your shortlist by 75% because the chances that they're going to share something together is only 25%. Okay, so it really becomes very effective. If it's consanguineous, it's a bit more tricky because if you've got parents who are related, you're going to get a higher number of homozygous rare variants. So it's a bit more tricky, but not impossible. What about autosomal dominant disorders? And here, your problem is that these people are going to inherit a lot of unimportant variants as well. So the most parsimonious design would be to sequence the most distantly affected individuals that you could, because these people will have the largest number of meioses between them, and therefore, by implication, the largest number of recombinant events. And you can calculate this out and say, what's the probability that we'd see this variant segregating with disease? And basically, if you've got n number of meioses between affected individuals, the chance that something's going to segregate is going to be 1 on 2 to the n. To the n, not 1 on 2n. And this family here, for example, if you were going to map this, this happens to be a Marfan's family, but you choose somebody down here and somebody over, well, not necessarily over here, but again, the number of generations away, the number of meiotic events, the less likely they are to have shared something by chance, and the more likely they are that if they are sharing something, that it's causative. You can also take sporadics, as we did for the MCTO case, and using unrelated individuals, and as few as 3 has resulted in being able to detect what the causative gene is. And in this case, you can look for the analysis of a single variant carried by all affected individuals or unique variants carried in a common gene. It really depends, though, on making sure that when you're saying you've got unrelated individuals, that they are actually all have the same disease. In other words, making sure you've got your phenotype right. However, one of the things that we've built into our pipeline is to allow for the presence of phenocopies. So if we've got 20 individuals who've got a disease, we can say, well, what about if we've got 15 of them sharing it or 10 of them sharing it? We don't have to assume that everybody has to be exactly the same. And you can also look for de novo dominant, looking at the children and the parents and looking at the number of novel coding variants in the offspring. Right. You need to have good capture. Paul's talked about that already, so I'm not going to talk about it. You need to make sure that what you're seeing is not artefact. And one of the most useful things that we've found over the last few years is having a large curated set, which enables you then to be able to say, this is just sequence error because of the way it's multi-mapping back, because of paralogous genes, because of pseudogenes. It's just sequence error. It really helps to be able to, if you're seeing something which is supposed to be only occurring in 1 in 100,000 people and it's coming up in 25% of your population, you know that that is not causative, that's just noise. You need to look at the population frequency of variants, and we go to a number of online databases, including DBSNP, Exact, 1,000 Genomes, and our in-host data. And this needs to be population specific. Many of our populations are Caucasian, but because of stuff we've been doing in motor neuron disease, we also are building a Chinese population as well, so that we have what's normal if you're Chinese. And also things have to be plausible, okay? And we have a look at that as well in terms of various protein prediction algorithms. So what about for diseases where we know what the gene is, but we want to type it more efficiently? And here we could say, well, could you capture all the known genes for a condition using an off-the-shelf whole exome chip? So you want to do this for, say, prohibitively large genes, or for conditions where you know that there's more than one known genetic cause. I mentioned already, OI has got 18, MODY has got, I think, 14 or so. FIOs are up to about 13 or 14% different types of genes. Or where you've got both problems. They're big genes and there's an awful lot of, there's a lot of them. At this point in the story, the current costs for library capture and next generation sequencing are lower than the commercial costs for sequencing most of these disorders. And I'll give you a comparison for a couple of these in a minute. At the moment, we send samples to 150 different labs worldwide, and could we all do this in-house instead? And the answer is yes. So this is Marfan syndrome. Now, Marfan syndrome you might think of as something you learn about at medical school, but the average age of death for a man was 40 and for a woman was 50, most of them dying due to ruptured aortic aneurysm. And this lad here, whose name was Florence Heiner, was a professional basketball player, and she died on the court in the middle of a professional basketball game. Volleyball. Okay, so she died. So things are a little bit better due to stringent blood pressure control and due to aortic root monitoring. And it's almost always due to mutations in fibrillin 1. The trouble is, it's a big gene and every family's got their own mutation. There's no hotspots. So generally, this is a clinically diagnosed condition. People don't do genetic testing because it's too expensive. And what we found when we looked at both Marfan's and we looked at osteogenesis imperfecta using the off-the-shelf exome chip, and we found 13 out of 13 mutations in OI and 9 out of the 10 mutations in Marfan's. We did find the 10th mutation, but it wasn't with sufficient confidence that we would necessarily have called it without having done further work. And the costs were about $800, and at that time it would have cost about $1,500 to $2,000 for fibrillin COL1alpha1 or COL1alpha2, each of them costing that much. So we published that in Bone Key, and we then went to do a proof of concept for FIOs or paragangliomas. Now these are neuroendocrine tumour cells. You will recall they secrete catecholamines. People can drop dead suddenly from crises of hypertension. And about 30 to 40% are familial, and in those, about 80 to 90% should have a germline mutation, and apparently sporadic cases. But even in the apparently sporadic, about 25% have a germline mutation. And one of the issues with FIOs is that penetrance is not perfect. You've got people who've lived to be a grandparent who have survived without having the disease, who've got the variant, and then you've got 5-year-olds presenting in the same family. So there's a lot of variability in penetrance in this situation. There are 15 susceptibility genes to date, some of which are regulating hypoxic response, and others in the RAS kinase-dependent growth pathways here. And again, we did a similar thing to have a look and say, well, could we screen all of these? Why would you want to do it? Because this is the most common heritable cancer. And the American College of Oncology says screening's warranted if there's a 10% chance of heritable malignancy. We know that overall, putting familial and sporadic together, it's about 30 to 40% in FIO, possibly 40 to 50% in FIO. So these really do fit what the guidelines are. We should be screening them. But at the moment, this doesn't happen in Queensland routinely. It doesn't happen. It's currently performed interstate, and they do one gene at a time, and it's done iteratively, and the turnaround time is years. So could we do this more efficiently? The answer is yes, you can. And we published this in Clinical Endocrine, and it was the editor's choice for January of last year, and we got an editorial as well. So can it be a clinically relevant and affordable test in a realistic time frame? Yes, it can. Those were proof of concept where we went to people we knew had a mutation and say we can pick it up. What about people we don't have a mutation? So we looked at short-rib thoracic dystrophies. We had 11 individuals without a known genetic cause, and we found the cause at the time. We found it in 10 individuals. We found the cause. We had one whom we didn't, but we've just found the last one. And we would have missed this one if panel sequencing was used, because the reason we found the last one was because we put it back on again and we came up with a result, and then we were talking to our colleagues and they had also found one. They'd also got an individual who had a variant there as well. So we actually would have picked up all of these people using exome sequencing. But because the panel wouldn't have had those genes on, we would have missed them because these genes weren't necessarily known at the time that we picked them up. How specific was it? Well, we went and had a look at our data from nearly 1,000 unaffected individuals and said, well, how often did we similarly see 2 persons with rare compound heterozygous variants of unknown significance? And we found 2 individuals out of those, which gave us a specificity of greater than 99%. So that's pretty good. 100% sensitivity and 99% specificity. You don't get many tests like that in biochem. And it costs about $850. So why would you want to do it? Because it makes a difference. And I want to use some examples, and this is absolutely fresh off the press, and you might have to get shot again. Maturity onset diabetes in the young, okay? So this is a type of diabetes which is not usually insulin dependent. Many of these people can use sulfonylureas, whereas in contrast, if you've got type 1 diabetes, you are, I see that camera, absolutely dependent on insulin. You're going to have to, I didn't realise about the video, we're going to have to change the video. Okay. 18 known MODY genes at the moment. And what we did here, in this particular case, we did do panel sequencing. We designed a panel, and we went off to WA. All children with type 1 diabetes are referred to this same database at Princess Margaret Hospital. And we had enough money to be able to do some of these, and we were able to get, we went to the cupboard, and the cupboard wasn't completely bare, but it didn't have quite as many as we'd hoped. We were able to get consent and DNA on 60% of the people who are antibody negative, and of 18 of the 64 patients who have allegedly had type 2 diabetes. And what we found was that 4 of the 60 of the antibody negative type 1s, these are people who've already been put on insulin. They've already been regarded as type 1s and put on insulin. So 5% of them have a MODY mutation, and there were another 13 that had been diagnosed clinically. If I just go back a page, this group over here, there were other clinical things that had made people think about it beforehand, and they decided they had MODY clinically and had kept them off it. Of those children who allegedly had type 2, a quarter of them had a MODY mutation. So if we just put this together in the limited amount that we've only been able to sequence a limited amount, but the minimum prevalence of MODY, if we put it in the whole database, the minimum prevalence is 2% of the entire clinic. Now, we've just received funding to be able to complete screening for the remainder of the database, including the antibody-positive type 1s. But the relevance is these children may be able to come off insulin safely. At a cost base, so they've done some cost analyses here. And is this cost effective to screen everyone? And the answer to that is, it depends how much it costs to sequence. If the cost for sequencing is $500 or less, it is cheaper to screen every child who presents with diabetes. I'll just repeat that. At $500, you would save money. It would be a net benefit to the health department to get people off insulin or to stop them going onto insulin, as well as the quality of life for the patients, etc. So if you sequenced every child with diabetes, you'd save money. So why does it matter too? Well, in the osteoporosis clinic, in my clinic, we went and had a look. We had 12 adults who we thought had a suspicious history. These were people who had not presented as kids. They weren't known to have OI. They were just people who had turned up with inexplicable osteoporosis, and a quarter of them had got mutations in known OI genes. If we looked at the completely unselected low bone mass cohort without knowing anything about their history, 6% of them look like they've got a form first of osteogenesis imperfecta. And the reason this matters is because conventional treatments for osteoporosis do not work well in adults who've got OI. So there's no point trying them. It's not going to work. You've got to think about something else. This is a higher prevalence than the prevalence of celiac disease, and yet we screen every adult with osteoporosis for celiac disease. So, just finishing. It's not perfect, and given the time frame, my husband just looked at his watch, I'm just going to say, I will be very happy to talk about this perhaps in question time. Things are not necessarily always perfect. You may not necessarily capture your gene of interest, although you can assess this previously. Some genes may not have been annotated. They may have only put one transcript in rather than everything. And there are technical challenges of capturing GC-rich sequences, which are common in the first exon. But that applies whether you do panel-based or whether you do exon-based. Panel-based do not necessarily capture exon 1 very well either. There are issues in terms of making sure that you don't get false positives. And you can sometimes get sequence similarity, which you really have to watch for the artifact. And I've talked about this again. Having a robust database to look for artifact is very important. What would be the benefits of whole exome sequencing versus whole GLM sequencing? This was a figure that was put up in 1995, and I suspect now most people would accept that this is actually an underestimation. But at least 85% and probably more are due to coding variants. If you're doing whole genome sequencing, you are invariably going to focus on exons initially. So given that it's going to be where you're going to find most of the information, you're going to throw away most of the genomic data when you're looking at monogenic disorders. In terms of the costs for coverage, Paul's already talked about this in terms of the relative cost for versus the depth of sequence for the different modalities. You need to have an excellent curated population exome variant database, things like the exact browser, to provide meaningful control data. And that's not available for whole genome sequencing at the moment, but it is available for exome sequencing. Both, by that I mean not just in-house data sets, but also things like the exact browser. It's much harder to attribute causality when you're looking at a non-coding whole genome sequence variant compared to when you're looking for something which is an exome variant. You need to make sure you've got a very high prevalence before you could say that something was statistically robust. And the platforms that we have at the moment make sure we've got very comprehensive and very even coverage of the exome, and that allows not just for the mutational events that I'm talking about, but also the structural variation that Matt talked about initially, such as CNV and exon deletion. Although WGS has got some advantages for gene fusion and for larger deletions, as we talked about in the cancers. What about panel versus WES? Well, you can design it to capture all of the exons, although again, first exons are usually tricky. Potentially, it's got lower costs for run, but that goes against the high initial costs of the custom platform design. You often have good depth of coverage across the targeted region. They can be quite rapid, they're pretty easy to analyse, and you don't have any risk of incidental findings. However, you've got no flexibility to go back to look at newly incorporated, newly discovered genes, and that means this is rapidly outdated. And Nadine went to a lecture last year where somebody was talking about this with non-syndromic deafness. By the time they got their panel, they only had 60% of the known genes because it's such a rapid rate of discovery. And you've got no post hoc discovery of going back to find new gene discovery in those cases which are negative for target genes. So, in monogenic diseases, genetic testing using MPS approaches, they can provide a meaningful, clinically relevant, affordable result in a realistic timeframe, which allows counselling for families and individuals, preventative intervention for at-risk individuals, such as myopia and paraganglioma patients, guidance of therapy, identification of novel therapeutic and preventable strategies. And this is relevant not just for the individuals, but also more broadly for society, because what we learn in monogenic diseases, we can apply to the general population. Thank you.